So thank you for joining us today. It's now my pleasure to introduce our speaker, Heidi Jones, Market Development Manager, Chromatography Applications and Collaborations from Bio-Rad Laboratories.
Thank you very much for that introduction, and thank you to the listeners for joining us today. Today we're going to be talking about effective strategies for endotoxin removal in therapeutic protein purification processes. First, I'll be discussing endotoxins and why they are so problematic in biotherapeutics. Next, I'll be discussing best practices to avoid endotoxin contamination, along with methods of endotoxin removal, and we'll end with some case studies describing the successful depletion of endotoxins using 2 of Bio-Rad's resins. So what are endotoxins? Endotoxins, formerly known as lipopolysaccharides, are complex components of the outer cell wall of gram-negative bacteria, such as E. coli, and are powerful stimulators of the innate immune system in mammals. Endotoxins are released in very high amounts during bacterial cell death as well as during mechanical cell lysis. They are designed to provide the bacterial cell walls with structural integrity and protection against invasion, and as a result, they are highly resistant to chemical attack and are extremely heat stable. Lipopolysaccharides are composed of 3 parts: the outermost component, the highly variable O antigen, which alerts the host immune system of invasion. Next is the core oligosaccharide region that links the inner and outer components together. And finally, there is a lipid A component that serves to anchor the lipopolysaccharide to the bacterial cell wall. The lipid A component is highly conserved and is the most toxic portion of endotoxins. Endotoxins vary widely from species to species and even amongst strains of bacteria, all of which contribute to their ability to persist and to their difficulty in removal from biological processes. So why are endotoxins so harmful, and why are we concerned about them in biotherapeutic drug production? While endotoxin is present in healthy human plasma at concentrations between 1 and 50 picogram per ml levels, at slightly elevated levels, endotoxins can activate the innate immune system, causing severe inflammatory responses. If endotoxins make their way to the bloodstream, the results can be septic shock and even death. Even low levels of endotoxin can cause a state of chronic non-resolving inflammation and have been implicated in various immunological and neurological diseases. Therefore, it is essential to ensure that endotoxin levels are low in final drug substances. Beyond the potential to harm patients, endotoxin contamination can result in significant processing delays and product recalls. Even at the discovery and research scale, the presence of endotoxins in biomolecules can have a negative impact, leading to false immunological readouts and conclusions. So how can we avoid endotoxins from getting into our products in the first place? The first line of defense against endotoxin contamination is actually just to ensure best practices in cleaning procedures. Endotoxins are everywhere in our lab environments, including, unfortunately, lab water sources. But fortunately, bleach and sodium hydroxide are both great decontaminants of endotoxins. And so by incorporating these cleaning agents into your standard cleaning practices and testing frequently for endotoxin contamination, you can significantly reduce the chances of endotoxin contamination in your subsequent workflows. If endotoxin contamination of your drug product does occur, it can be extremely challenging to adequately remove it, as you may have already experienced and might be the reason why you are listening to this webcast. The reason for this has to do with the very function of endotoxins. They are designed to withstand destruction. They are generally negatively charged at pHs above 2 and tend to form superaggregates in solution. And on top of all of that, they interact in unique ways with different proteins. So as a result, there isn't a singular solution for endotoxin depletion. Nonetheless, there are various attributes of endotoxins that can serve as clues as to how they can be depleted, including their potentials for various interaction types, such as hydrogen bonding, electrostatic interactions, metal affinity, and hydrophobic interactions. Fortunately, a variety of methods exist for the removal of endotoxins from biological solutions. When choosing a method, considerations include maintaining biotherapeutic activity and maximizing product yield, which become even more critical at larger production scales. Effective methods of endotoxin depletion include those that manipulate electrostatic interactions and the negative charge of endotoxins, such as ion exchange and mixed mode chromatography. While LPS affinity resins like polymyxin are popular at research scale, they are cost prohibitive at process scale and may reduce yields if proteins closely interact with endotoxins. Size-based separation techniques like ultrafiltration or size exclusion chromatography yield mixed results depending on endotoxin aggregation levels relative to the protein. Concentrating endotoxins during removal methods can lead to even more challenging situations. At process scale, resin-based removal methods, especially ion exchange and mixed-mode chromatography, are standard and effective due to their manipulation of electrostatic interactions and the net negative charge of endotoxins. Anion exchange chromatography is particularly well suited for endotoxin removal from acidic proteins. Depending on the buffer system used, anion exchange resins will bind endotoxins tightly while the protein flows through during the load or elutes at low salt concentrations, minimizing protein yield losses. Bio-Rad offers several strong anion exchange resins that have been validated for endotoxin removal, including MacroPrep High-Q, Nuvia-Q, and Unosphere-Q. All three of these resins share a quaternary amine ligand built on different matrix supports. MacroPrep High-Q, which is built on a rigid macroporous methacrylate polymer matrix, has a high ligand density and is particularly well validated for its ability to remove high levels of endotoxin, even with strong protein-endotoxin interactions. On the next few slides, we'll look at a couple of case studies featuring MacroPrep High-Q. In this first case study, a panel of supports was evaluated to identify the top-performing support for satisfactory endotoxin removal from protein X, a small acidic protein produced in yeast. 20 supports were initially screened. 6 of these supports were selected for further evaluation. These supports were equilibrated in PBS, pH 7.4, for endotoxin polishing of protein X, which had been previously passed through another strong anion exchange column operating in flow-through mode. MacroPrep High-Q strong anion exchange support was chosen for the endotoxin polishing step because it resulted in the lowest ratio of endotoxin per milligram of protein X. MacroPrep High-Q was then scaled up to process the remaining contaminated protein X material, resulting in an almost 3 log reduction of endotoxins with a step yield of greater than 99%. MacroPrep High-Q's high ligand density, macroporous structure, and possibly slight repulsion of lipid A from the surface of the bead due to the bead's slight hydrophobicity may have enhanced endotoxin binding to this resin and separation from the protein. This next case study demonstrates that MacroPrep High-Q can successfully remove endotoxins from a small, very acidic protein exhibiting a strong affinity for the lipid A portion of endotoxins. This protein, referred to as protein Z, had a very high starting endotoxin value of 41,000 EU per meg. After applying protein Z to a MacroPrep column equilibrated at pH 7.2, it was observed that at low salt concentrations, 20% of the active protein Z was eluted, while highly concentrated endotoxins eluted at higher salt concentrations. Based on this observation, and given the affinity for lipids observed with this protein, subsequent optimization work found that by denaturing protein Z, interactions with the lipid A portion of the endotoxin was significantly reduced. This resulted in the majority of protein Z eluting in the flow-through and a 99.9% depletion of endotoxins. MacroPrep High-Q's macroporous structure and high ligand density likely enabled increased electrostatic interactions between the resin and the endotoxin while enabling the dissociated protein Z to elute in the flow-through. Another useful tool for endotoxin removal is multimodal or mixed-mode chromatography, which combines 2 or more modalities of interaction, such as ion exchange, hydrophobic interactions, size exclusion, and hydrogen bonding, into one resin or membrane support. Bio-Rad offers several resins that have been successfully demonstrated to deplete endotoxins from biotherapeutic solutions. These include ceramic hydroxyapatite media, or CHT, featuring metal affinity and cation exchange, along with Nuvia C', a weak cation exchanger and hydrophobic interaction resin, as well as Nuvia A' 4A, a strong anion exchanger and hydrophobic interaction resin. Mixed-mode chromatography interactions are not independent of one another. For example, when using a mixed-mode ligand containing both hydrophobic and ionic elements, increasing ionic strength will disrupt ionic bonds. However, increasing salt concentrations will promote hydrophobic interactions. Next, we'll take a closer look at how CHT can be used to deplete endotoxins from biotherapeutics. Ceramic hydroxyapatite, known as CHT, is a mixed-mode chromatography media used for the purification of a variety of proteins, including monoclonal and polyclonal antibodies, antibody fragments, enzymes, nucleic acids, and membrane proteins. It is formed from the chemical synthesis of calcium and phosphate salts. Biomolecules can interact with CHT through either calcium affinity interactions and/or cation exchange interactions. Due to their highly acidic nature and high levels of carboxyl and phosphate groups, particularly in the core and lipid A regions, endotoxins have a high affinity for CHT, interacting primarily through metal affinity interactions, modulated by either classical polydentate coordination in the case of carboxyl clusters, or through modulation with phosphoryl oxygens in the case of phosphoryl residues. In this first case study, buffer optimization was performed to tease out separation of a monoclonal IgG antibody from high levels of endotoxin contamination. The antibody and endotoxins co-eluted when a phosphate gradient was applied, while the endotoxin eluted separately when a sodium chloride elution gradient was applied, while bound endotoxins were eluted in a high phosphate strip. This result demonstrates that the primary mechanism of interaction with endotoxin was through its carboxyl and particularly its phosphoryl group and the charged calcium sites on CHT. It has been observed that very slight increases in phosphate concentration and sodium chloride gradients increase the level of endotoxin slightly in the elution pool, indicating that there is also a smaller cation exchange component to endotoxin's interaction with CHT. In this particular case study, endotoxin was completely depleted from the antibody to levels below detection. The second case study showcases CHT's unique ability to remove very high levels of aggregate, along with other impurities and endotoxins. In this case, a mAb with 34% aggregate after the protein A elution step was loaded onto the CHT column that had been equilibrated in 10 millimolar sodium phosphate pH 7. A gradient elution was applied from 0 to 1 molar NaCl in the 10 millimolar sodium phosphate buffer over 20 CVs. The resulting mAb pool was 97% purified monomer with simultaneous reduction in various impurities, including endotoxins, which in this case were reduced from 11 EU per meg to 1 EU per meg. To summarize the benefits of CHT, it is a very effective resin for the removal of multiple process stream contaminants, including leach protein A, host cell proteins and DNA, aggregates as well as endotoxins, due to its dual mechanisms of action through both metal affinity calcium binding sites and phosphate residues. In antibody production workflows, CHT can be used as part of a two-step process following affinity chromatography, and can be incorporated in place of anion exchange in three-step processes, given its ability to clear impurities. CHT can also withstand high pressures and flow rates, making it an efficient part of any purification workstream. While primarily used as a polishing resin, CHT has the capability of being used as a capture resin for a variety of recombinant proteins and enzymes. CHT is available in pre-packed forms, ranging in size from 1 ml to 17-liter pre-packed columns, making them easy to plug and play into continuous processing workflows. To summarize, endotoxins are common and significant pyrogens. They are designed to avoid destruction, making them especially difficult to remove from biotherapeutics. However, there are a number of methods that you can use to reduce their impact in your processes. Preemptively taking standard cleaning practices up a notch, along with frequent testing, can significantly reduce contamination events. For removal of endotoxins from drug substances, anion exchange and multimodal chromatography work well and are scalable. Bio-Rad offers a selection of both that have been shown to reduce endotoxin levels. In this webcast, we examined MacroPrep HiQ for removal of endotoxins from acidic as well as lipophilic proteins. And we also looked at CHT, ceramic hydroxyapatite, for removal of endotoxins from antibodies. Both are cost-effective choices for large-scale bioprocessing. Bio-Rad also carries AffiPrep polymyxin resin that can be used at research scale for endotoxin removal. So with that, I'd like to say thank you for joining us today for this Ask the Expert webcast. Please don't hesitate to contact us if you would like to discuss any of the material discussed here today in further detail. And please visit our digital resource hub, where you can request free resin samples, speak to our scientists, and more. So with that, thanks again, and I'm happy to take any questions.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for Heidi now. So our first question is, why should we be concerned with endotoxin when dealing with mammalian cell productions?
Great question. Endotoxin contamination is problematic for all production types. This is because endotoxins can be found on all lab surfaces, in media, glassware, consumables, buffers, and is especially prevalent in our lab water sources. Therefore, it's really important to test for endotoxin levels often. An in vitro cell culture study, actually, their presence has been shown to affect cell lines as well as cell line growth. And this is especially true for cell lines involved in the innate immune system, such as monocytes and macrophages. And adding to that, in downstream processing, when you think about how purification workflows usually involve multiple column steps, you can see how levels of endotoxin at each step, even if they're very, very low, can build up. Hence the importance of stringent cleaning procedures and regular testing at various stages of a purification process. And why it's also good to incorporate endotoxin depleting resins, such as the ones described here, into purification processes.
All right, yeah, and our next question is, are these resins scalable for endotoxin removal?
Yes, actually, Bio-Rad's chromatography resins are indeed scalable for endotoxin removal during protein purification. These include the ion exchange and mixed mode resins that we've discussed today. They're available in as small size as 1 ml up to 17-liter pre-packed in the case of CHT. So if you would like to receive a sample of these resins, please email us at process@biorad.com.
All right, well, it looks like we're running just a bit short on time, so if we don't get to your questions, we'll be passing them along to Heidi, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, what are purified endotoxins used for?
That's a great question. Actually, purified endotoxins are used for a variety of purposes, such as in research, in diagnostic kits, as well as in assays, such as sepsis diagnosis and quality control. Purified endotoxins are also used to explore endotoxin and removal methods, such as in the example of CHT described here. Additionally, in biotech, researchers are investigating alternatives to commonly used expression systems such as E. coli through modification of the endotoxins. So here would be another use for purified endotoxins serving as controls in assays, for instance.
All right, well, thank you so much, Heidi.
Thank you.